everyone. Thank you for joining today. My name is Gus Bauman, and today we're going to be going over some practical guidance on putting BeCrop technology to work, specifically with a focus on using soil intelligence to inform seed selection. So I think an interesting topic, a lot of talk when it comes to soil biology testing with biostimulants and biofertilizers and products of that sort of segments, but today we'll be talking seed selection and seed treatments, specifically with a focus on practical applications of Biomaker's BeCrop technology. Before I get any deeper into an introduction, just going to wait another minute or two because I see we still have a lot of attendees trickling in. So just pause for a moment here and let folks join, and in a minute or two we'll go ahead and get started, get a proper introduction, and cover some housekeeping items. All right, looks like our attendee numbers are still going up. So again, just going to wait maybe another 30 seconds to a minute to let folks join, and then we'll kick things off on this webinar focused on using soil intelligence to inform seed selection. All right, well, I think we're all set to go ahead and get started. I think some folks might still be trickling in, but I'll go ahead and get us kicked off with an introduction. Thank you all for joining today. Good morning, good afternoon, good evening, depending on where you all are in the world. Thank you for tuning in, and I'll be talking today about sort of an interesting angle to BeCrop technology. And for those who are new to BeCrop and Biomakers, we'll be going over just a brief synopsis of what BeCrop technology entails and soil biology testing in general. But a majority of this webinar will be focused on a practical angle with an emphasis on how BeCrop technology can be used to inform seed selection, particularly in row crop applications, but really these case studies we're going to be going over today apply pretty broadly across different crops, different agricultural systems, different regions. And I'll go ahead and just kind of do a really quick intro to our, looks like, oh, there we go. I just got to make sure my slides are changing. So do a quick overview of our agenda today and then cover some housekeeping items. So we're going to start with an introduction and an overview of BeCrop, particularly BeCrop Farm, which is Biomaker's newest service offering. I'm going to just give a synopsis of common seed traits and treatments on the market, just to kind of tee up what sort of angles BeCrop can be used to inform seed selection, and then go over really the integration of soil biology data into the seed selection process. So just kind of the theory and process behind taking soil biology data into actual seed selection. Very pertinent this time of year, and I know that a lot of seed purchase decisions are sort of being pushed out, partially due to market conditions. And then we'll be covering some specific case studies, going into just some examples using real BeCrop data on how that data can be used to inform seed choice, make decisions, not just based on some of the standard influences on seed selection, but going a little bit deeper into how soil health and soil biology can be used to make optimal decisions as far as seed. And then we'll finish with a question and answer session. I'll be aiming to save maybe 15 minutes or so for questions. So with that being said, just want to cover a couple of quick housekeeping items. First of all, related to the Q&A, feel free to submit questions at any time throughout the webinar in the chat, and we'll try to get to as many as I can in that last 15 minutes or so. Any questions that I'm not able to answer, we'll be sure to reach out over email and get those questions answered, or connect you with someone on the Biomakers team. And then lastly, this webinar will be recorded and sent out following the conclusion of the webinar. So if you have to hop off early, or if maybe you weren't able to join, we'll be sure to get the recording out so that you can view it at a later date. So with that being said, we'll kick things off and start out with a quick introduction and overview of BeCrop and BeCrop Farm. My name is Gus Plowman. I am a certified crop advisor and work as a soil health and regenerative ag consultant. I work as an agronomic consultant for Biomakers, and generally what my work is focused on is expanding the adoption of regenerative ag principles, as well as regenerative ag and soil health tools, technologies, and inputs that can help support the growth of farming systems that really farm in harmony with nature and focus more on integrating aspects like biology into agronomy. So I've had quite a bit of experience working with BeCrop technology over the last 4 years or so. And this slide will just kind of give a really quick overview of BeCrop, which for those who may be on this call and aren't familiar, is a soil biology testing service. And what BeCrop technology is really underpinned by is a massive soils database housing tens of millions of soil microorganisms. I believe that the current number may even be higher than 55 million listed here. Using that expansive database, as well as computing and AI-based technology, Biomakers provides soil biology testing that can provide practical insights on agronomic decision-making. So while soil microbiology as a whole can be a really complex field with a lot of angles to look at, like environmental remediation, geology, all sorts of disciplines that are sort of intertwined when it comes to soil, because soil really has an impact on a lot of different aspects of human civilization. What Biomakers technology is really focused on is providing insights relevant to agronomy. So focused on some of the ways that soil biology, bacteria, fungi, and other microorganisms influence farm outcomes, influence what practices really lead to the most sustainable outcomes and productivity for farmers across the world. BeCrop technology has been used in over 30 peer-reviewed publications and is available across 6 continents and over 56 countries. Some of those outcomes that I mentioned, so again, BeCrop technology is really honed in on the agronomy side of soil, which I think is really one of the angles of which the name BeCrop came from, looking at soil biology from the perspective of the crop. So some of those areas that BeCrop supports with is predicting disease risk and crop stress tolerance or stress adaptation, informing nutrient cycling, health and biodiversity. Some other services Biomakers offers includes the ability to measure and quantify agricultural input performance, monitor farm practices over time to determine how they're impacting the soil microbiome, measure and verify sustainability outcomes. And then lastly, while again, BeCrop technology is focused on practical aspects of agronomy, the technology can also be used to provide raw data on the soil microbiome for research. So to give a really quick overview of how this technology works, soil is not necessarily just an inert substance, or just essentially dirt or a media for life for plants to grow in. There's also a massive ecosystem of soil microbes, potentially millions to billions of cells of bacteria, fungi, other microorganisms in just a spoonful of soil. So that symbiotic ecosystem, while we can't necessarily see it, really does have a significant impact. There's a lot of research that's come out over the last 10, 15 years on the influence that the soil microbiome has on food quality, on farm management. So the way that Biomakers technology works is that it starts with with soil samples, and from those soil samples, they undergo a process of extracting and measuring and decoding the DNA, the genetic material that's in that soil, using specific biomarkers that certain soil microorganisms have. So that's this first step from soil to biodata. So that soil is decoded, the microbes are identified based on their taxonomy, but of course a long list of the soil microbe names, their taxonomy might be useful data to some really savvy folks who have tools and background in soil microbiology. But to anyone, including myself, it's really not going to tell you a whole lot of concrete or practical information just using that taxonomic information on soil microbe identity. So that's where this second step of the process comes into play, which is when that biodata is then converted using AI-driven bioinformatics process that BioMakers offers into a functional analysis report. So that's taking the species and genus level data on soil microbe identities, running that through a bioinformatics technology system that is able to profile the microbial functions of those species, and then ultimately the output of a soil test is a functional analysis report that doesn't just give species identities, but translates that into the actual agronomic related functions that those microbes perform. Again, like nutrient cycling, disease risk levels, microbes that produce phytohormones, and general soil health indicators like biodiversity. So this is really, I'd say the second step of the process is really the value proposition that BioMakers offers. There's plenty of labs out there that can do the genomic sequencing and take a sample of soil or other material and measure the microbes present with sequencing, but then being able to profile those microbes and also provide functionality scores to give you an idea of what their relative level is, compared to soils, across the world. That's really the value in BeCrop technology. And so this is a tremendous simplification of the analysis process, but really it's that initial soil to biodata step followed by the microbiome functionality profiling step that really provides data that can be used by farmers, retailers, ag input providers, agronomists, crop consultants, really anyone who's informing agronomic decisions and ultimately trying to provide more insights on what's going to most effectively promote soil productivity. So with that, just another sort of quick overview of BeCrop. These are sort of the three main foundational categories that BeCrop data falls into: soil quality, plant health, and nutrient cycling. Soil quality really encompasses the more overarching aspects of the soil microbiome. So rather than honing in on like specific functions like nutrient cycling or disease, soil quality sort of looks at the entire population, from a big picture perspective. So this includes metrics like biodiversity, telling you, how many different species are there, how even are they, is there a wide range of species that are very different genetically, or is there sort of a relatively uniform, not very biodiverse population? Overall functionality and the resilience of the microbiome, how effective it is at resisting change or stressors. So this can provide some information on how susceptible the microbiome might be to stressors like tillage, chemical fungicide inputs, and fumigants. And then fungi to bacteria ratio and mycorrhizal ratio, just informative indicators that give an idea of how the overall structure of the microbiome looks. Obviously, with a lot of soil biology tests out there, including PLFA and others that have been around a little bit longer than soil genomics, these kind of follow the same sort of use case of really just giving you an idea of how the balance between fungi and bacteria looks. In most cases, healthier soil or soil that has a more functional ecosystem has a stronger balance of fungi. And then mycorrhizal ratio just really tells you if there's mycorrhizae present and whether or not there is a balance between endomycorrhizae and ectomycorrhizae, which have different niches living on or partially inside of the plant root. Again, just informative indicators don't really, and I say informative indicators because they don't really get into the specific functions that microbes perform, which are covered more in the next two categories. But again, soil quality, this category really just gives a rundown of what the overall soil health status and status of the soil microbiome is, to really see whether or not maybe soil is moving in the right direction or whether there's a lot of room for improvement across the microbial population. And then plant health, this is really looking at more specific functions of soil microbes. So any soil-borne diseases, which are flagged according to crop type, are shown here with a risk index. natural biocontrol agents. So we're going to talk about that quite a bit today, the beneficial microbes that can suppress fungal pathogens, bacterial pathogens, insects, and nematodes. So these natural biocontrol agents are sort of like your soil's natural disease suppressive qualities, microbes that can feed on, predate, or parasitize pathogens or pests that are present. And then phytohormone producers and stress adaptation metabolite producers. These are the microbes that are present in the soil that I would define as plant growth promoters, because they're able to produce and donate beneficial PGRs, plant growth regulators like auxin, cytokinin, ACC deaminase. And by producing these plant hormones, they're able to help those plants deal with stressors, help plants have more vigorous root growth, nutrient uptake, deal with factors like soil salinity and drought. So we'll be touching on that too in relation to seed and seed treatment selection. And then lastly, nutrient cycling. So this is a category that looks at all the different microbial functions involved in unlocking nutrients, converting them between different forms, making them more plant available. So of course, NP and K cycling are among the primary focus of BeCrop. Carbon pathways, which includes microbes that emit carbon from the soil through the process of aerobic respiration and anaerobic processes. So looks at both microbes that move carbon into the soil, the microbes that are responsible for carbon sequestration, as well as the microbes that export carbon. And then organic matter breakdowns, another key and practical component of that, looking at the microbes that are able to break down crop residue, composts, even organic materials like insect carcasses that we don't really think about, but can provide some nutrient and carbon contribution. This measures the microbes present that can drive that process of converting those organic matter sources into stabilized carbon, as well as releasing other nutrients in the process. And then lastly, micronutrient pathways, the microbes that cycle some of those important micronutrients. And a lot of micronutrient deficiencies can be really complex, influenced by soil type, texture. The bedrock of the soil can have a big influence on what micronutrients are going to be prevalent, which ones are going to be locked up. But soil biology is also a really important component there, because we know that a lot of microbes do play important roles in both using and also releasing micronutrients in plant available forms. So that's another key element to kind of add another angle to informing how to get to the root cause of nutrient deficiencies. And then lastly, BeCrop also includes some optional chemical fertility tests offered by Biomakers Partnership Labs. This includes the Haney soil health tests, your classic Malick 1 and 3 soil fertility tests, and a total soil digestion, which can look at the overall nutrient profile of the soil. So these chemical fertility tests can serve great in conjunction to give you both the chemical profile of the soil in terms of nutrient availability, along with the biological profile based on the BeCrop test. So now just an overview of the specific solutions that Biomakers offers. This includes BeCrop Test, which is just standalone single soil sample assessment of soil biology. Then BeCrop Farm, which we'll touch on a little bit more in a minute with our case studies. This is essentially soil biology data provided on a precision ag level with agronomy insights and recommendations, as well as a mapping program to look at how soil biology and microbial populations vary across the farm at the field level and subfield level. And then we also, the third service that Biomakers offers is BeCrop Trials, which is replicated trials designed to look at inputs as well as untreated controls over time to measure crop input performance and determine exactly how inputs are influencing the soil microbiome. So that's more with a focus on understanding products and R&D, as well as putting together claims and valid understanding of how inputs like biostimulants, fertilizers, crop protection products are influencing the soil microbiome. So BeCrop Farm, which we'll be using for the use cases on seed selection today, the way that BeCrop Farm works is, aside from just looking at individual tests or sets of tests, BeCrop Farm integrates a range of remote sensing data sets like NDVI, looking at satellite-based insights on vegetation levels in the field, soil texture, soil moisture, soil wetness, or some other factors that are known to influence soil microbiology. And then these different layers of remote sensing data are used to inform and target where soil tests can most effectively deliver data on representative zones. So that's what's used to ultimately create these soil biology maps that are akin to, say, grid sampling, precision ag soil fertility maps. A little bit more detail on how that's done, and I'm not going to go too deep into detail here, but this basically is just an overview of how those satellite layers are looked at. This starts with a field shapefile or boundaries that are uploaded by a Biomaker's client. Once that map of the field is uploaded, Biomaker's automatically analyzes all those variables that I mentioned, like NDVI, soil texture, soil type, soil moisture, and those are all known to have a heavy influence on shaping and selecting for biological soil microbe populations. So that data is used, to predict which biological zones are representative and need to be sampled in order to map out the soil biology across a field. So once Biomakers receives those boundaries or shapefiles of the field, it's used to develop these biological zones, and then sampling locations are shared back with the client, those specific GPS coordinates for a sample, so that clients know exactly where to sample in order to generate those high definition or precision ag maps that can show you how soil biology varies within each field and across fields. So looked at in another angle, or really just kind of another aspect of BeCrop Farm, aside from just looking at field by field or farm by farm data. Once those soil samples are submitted, the sets of soil samples create field and subfield level sampling zones, and then this can be organized across a farm, multiple fields within a farm, and also multiple farms within a client's account within the BeCrop portal. So a retailer, or say a crop consultant, someone who's managing data from multiple farms, can sort of segment out those farms so that they can provide access to individual clients to just see those clients, their own data, but also be able to organize that data on a client or regional level so that they have access to everything, but they're able to again toggle that access to individual clients so that that data is nice and organized. So allows for data to be looked at and different angles, and not just necessarily looked at on a farm by farm basis. So now to get into our topic today, agricultural seed traits and genetics. And we're just going to start with a quick overview. And generally, I know genetics and seed traits, sometimes those terms are used interchangeably. Genetics really refers to the genetic profile or the potential of a seed, whereas traits refers to some of the individual functions or features of a seed that can be either genetically engineered and added to a seed's genetic profile, or in some cases selected for through breeding. We're just going to kind of use those terms together to mean overall seed hybrid features and traits. But obviously, just wanted to acknowledge that there is a distinction when we talk about the terms traits and genetics. So this is definitely not a comprehensive list, but just an overview of the top seed traits on the market, and also seed treatments. So we're going to talk seed treatments today as well. As far as seed traits and genetics go, herbicide resistance, probably number one out there, or I should say number one in the row crop space, particularly with crops like corn and soybean. Insect resistance, so specifically the Bt trait used to confer resistance to insects, is also probably up there as one of the major traits on the market. Disease resistance, or I should say disease tolerance, is another big one. Crops that are able to maybe not always be 100% resistant, but able to tolerate certain microbial soil-borne pathogens or foliar pathogens. And then nematode resistance kind of goes hand in hand with that. Not all nematode resistant hybrids are going to be entirely resistant, but maybe more or less tolerant of certain plant parasitic nematodes. And then stress tolerance is another really big one out there on the market, especially in regions that are maybe more drought stress or deal with big gradients in temperature and other environmental stressors. So lots of seed hybrids out there that are able to basically deal with some of those stressors. And these are all going to be things that we kind of explore how BeCrop influences. As far as seed treatments, I'd say these kind of fall into three main categories of crop protection. You know, your conventional fungicides, insecticides, and nematicides. Then you have biological crop protection seed treatments. These are going to be your seed treatments that contain some of those beneficial biocontrol agents. We will refer to them as biofungicides, bioinsecticides for the beneficial microbe inoculants that can suppress insects, and then bionematicides for the microbes, the microbial seed treatments that can suppress nematodes. And then biostimulants, microbial seed treatments that help enhance nutrient cycling, can promote stress tolerance of the crop, and in some cases promote root growth, and really help give a seed a head start by placing microbes or biostimulant compounds that can give that seed sort of a leg up in helping its root growth, helping its early vigor. So it's a really this is just a few bullet points here, but this can really get pretty expansive when you start to break down all the different combinations of these seed traits and seed treatments that can go out. This is just, again, this is not a comprehensive example, but just a general example of the different seed traits and treatments that a retailer might have in their portfolio. And this when you think about all these different traits, herbicide resistance probably number one, then insect resistance pretty prevalent on the market in traded seeds, foliar disease resistance, seedling disease resistance, nematode resistance, or I should say tolerance, abiotic stress tolerance, high yield potential. And then you also, a lot of retailers are often offering stacked seed combinations that might have herbicide resistance, insect resistance, some degree of disease tolerance, and or stress tolerance. So, there can be two-way, three-way, maybe even four-way stacked combinations of seed traits. So there's really, when you really break it down, there's going to be hundreds, if not thousands, of different combinations. And then you layer in seed treatments over here on the right, biofertilizers that focus on nitrogen cycling, mycorrhizal seed treatments, stress tolerance promoting seed treatments, some of those biological seed treatments, and then of course probably the most prevalent are conventional fungicide, insecticide seed treatments. Obviously, these combinations start to get well into the thousands, and it's there's factors out there that can help determine some of these selective some of these make some of these decisions and ultimately weed out what we're looking at. But soil biology is one aspect that traditionally has not really been a part of the seed selection process. There's been obviously some of these ways that observations on soil biology, like observations of soil-borne disease, can go into informing disease-resistant or disease-tolerant hybrids and seed treatment. But ultimately, I think a lot of that comes from just knowing what diseases have been prevalent and problematic in past seasons, and then just sort of making an assumption about what's going to be an issue in the upcoming season. So just to really quickly connect some of the BeCrop soil biological metrics that relate to these different seed traits and seed treatments. Really, we're going to be looking pretty heavily at biocontrol agents, insecticide agents, fungicide, nematicide agents, as well as the disease risk indicators on the BeCrop report, and then stress adaptation and plant growth promoters or plant growth regulators. So the little line here just kind of connects some of those seed traits we were just looking at with the BeCrop metrics that are ultimately going to inform the selection of some of these seed traits. And it's pretty straightforward. Obviously, you talk nematodes or insects, then insecticide agent, nematicide agent are going to be kind of main areas of focus. And then to get into seed treatments, the combinations get a little bit more complex here with crop protection, seed treatments, obviously disease risk, and some of those beneficial biocontrol agents, insecticide agents, nematicide agents are going to influence, how much, to what extent a field really can benefit from a crop protection seed treatment. Biocontrol, biological seed treatments, really the same story there. Biofungicides, bioinsecticides, bionematicides, you may not need them if you already have really high levels of beneficial biocontrol agents helping suppress diseases or pests in the soil. And then biostimulants, and that includes biofertilizers, nutrient cycling microbes, mycorrhizae, stress tolerance promoters, root growth promoters, those stress adaptation and phytohormone producing microbes really are going to, ultimately dictate whether or not that's maybe a yield limiting factor or not when going to select a seed treatment. And then lastly, all three of these categories you can see also connect to biodiversity and overall soil health. So it's really, there's a lot of complexity to sort of demystify when it comes to making decisions about ag inputs and how they influence just overall soil health, whether they maybe have some unintended negative consequences on hurting soil health or reducing microbial biodiversity. And having just an overall idea of what the biodiversity or what that soil microbiome looks like can give you an idea of whether or not to prioritize, biological, I'd say I'll define them as biosolutions, biostimulants and biological inoculants, or, maybe in some cases you do have soil that is, really functioning at a high level, and maybe if you have a significant issue, then conventional crop protection products can be a valuable tool in the toolkit. But at least with looking at biodiversity and overall soil health indicators, you're able to get an idea of how susceptible that soil is, or to unintended consequences of conventional crop protection products, or just whether or not they have a significant impact on that soil microbiome, or maybe they don't really have a significant impact on the soil microbes that are present. So traditionally in gray here, we have, based on a seed retailer's portfolio, these are some of the factors that I think are commonly used to dictate and determine what seed traits and seed treatments are used. So herbicide program, probably first and foremost, obviously going to determine if a grower is using herbicide resistant seed hybrids. And then history of disease, pest, and nematode pressure. So again, based on sort of the assumption that issues in the past are going to pop up in the future, although this obviously can vary year to year based on environmental conditions. History of abiotic stressors, again, pretty speculatory. Some years there may be significant drought, other areas not. Some places are pretty drought prone. Other regions you might have drought pop up, every 5 or 10 years rather than every other year. And yield expectations also, again, kind of speculatory, but is something that influences some of those, high yield potential hybrids. And then highlighted here in gold are the agronomic factors that BeCrop can provide. So quantifiable, measurable soil-borne disease presence and risk, beneficial biocontrol agents, so you can determine the soil's natural ability to suppress, fungal diseases, insects, and nematodes. Beneficial stress tolerance promoting microbes. So another metric that can layer in with the actual history of abiotic stressors. Do you have microbes there that are already helping the crop deal with those abiotic stressors, or is this something to prioritize with a seed treatment or specific trait of a seed? Beneficial nutrient cycling microbes. So again, if you do have strong nutrient cycling, maybe prioritize on crop protection. But if those nutrient cycling microbes aren't there, then of course a seed treatment that maybe inoculates nitrogen fixers or phosphorus solubilizers is really key. And then again, overall soil biodiversity and soil health. Some of those indicators, just to give you an idea of, is the needle moving in the right direction? Are conventional products maybe having a negative effect on overall soil health, or is soil health moving in the right direction, to just get an idea of whether or not you should prioritize inputs, products, seed treatments that can support soil health, or if maybe things are moving in the right direction, start to hone in on specific action areas. So now to go into our case studies. So we're going to take some of, I know time is kind of flying by, so I'm going to try to go through these case studies pretty quickly and try to stay informative. Again, feel free to pop any questions into the chat that we'll answer in the last 10-15 minutes or so. So I'm going to take some real BeCrop Farm data and basically go through just some examples of how that real data can be used to inform seed selection across three adjacent fields. So again, going back to, this is that same slide from before on just an example of the seed traits or traded seeds and the seed treatments that might be in an ag retailer's portfolio. And again, when we look at all the different combinations, the stacked traits, the different seed treatments that might all be mixed in together, there's hundreds, if not thousands, of different combinations to look at. So aside from just those speculative, information that, based like disease from past seasons, herbicide use, insect prevalence year to year, abiotic stressors, how can we use real-time BeCrop Farm data to start to prioritize which of these traits and seed treatments make the most sense to optimize crop outcomes. So first, we'll take a disease example because this is probably the most straightforward. And we're looking at BeCrop Farm. We're looking at 3 different fields labeled. This is field 1 in the bottom right, field 2 here, and field 3 in the top left. And this is the disease risk index for fusarium scored across cornfields. So the scoring index ranges from very low levels in the yellow to very high levels in the dark red. That's based on the risk of fusarium. And again, that's informed by the actual levels of fusarium pathogens in the soil, as well as other factors that can influence fusarium impact on the crop. So starting with field 1 down here, it has a maximum disease risk score of 94, which is in that very, very high level, 80 to 100 BeCrop score range. That's again the max, not the average, but you can see most of this field is in the red, and this little histogram breakdown here looks at the total percentage of acres in each disease risk level, and you can see a majority, 60%, it looks like, of the field is in that high level, a 60 to 80 percentile ratio when compared against BioMaker's database. So this field is quite high risk, whereas field 2 and field 3, just kind of looking at the color-coded index, majority of these fields are in the low range. So that's the 20 to 39 percentile range when reflected back on the BioMaker's database. So there's definitely some fusarium present, a couple spots along the edges that are showing a little bit higher in the medium to high ranges. But really, field 1 is the field to prioritize those, any fungicide or biofungicide products. Or I don't know if there's a lot of seed varieties out there that have significant resistance to fusarium, but I know there are some hybrids that have fusarium tolerance or are able to produce early vigorous growth to kind of get ahead of early seed disease infection. So field 1, this would be a case where there's a high need, or the conclusion here is to prioritize those fungicides or biofungicides that make claims of treating fusarium in that field. Field 2 and 3, moderate need. The maximum scores are in the 70s, but generally across most of the field, fusarium risk is in the low ranges. So that's going to check the box for field 1 as far as placing fungicide or biofungicide seed treatments and fusarium tolerant seed varieties. Now looking at another angle of disease, this is levels of beneficial fungicide agents. So these maps are measuring the microbes that are able to suppress fungal pathogens like fusarium. So we can see, and this is scored on a different index than disease. So because we're looking at beneficial microbes, you'll see in the top right our legend here. It's scored from very low levels in the yellow to very high levels in the dark blue based on BioMaker's corn database. And you can see majority of these fields are in those blue ranges from medium to very high. Fields 3 and fields 2 here on the left have a lot of dark blue, a lot of ranges that are very high, very little yellow. Field 2 has a little yellow spot here, but on average, these fields score 66 percentile for field 2, 72 BeCrop score index for field 3. So these are really scoring in that upper 30, 40% of samples in BioMaker's database, whereas field 1 here scored a 59, so it's only a little bit above average. And this sort of northwest quadrant, or I'd say northwest third here, is more in the medium range with a few yellow spots. So this field scored on average lower than the other two, and it looks like there's sort of a significant stretch of the field that's scoring really in an area where it's not super low, but it could use improvement. So the conclusion here, field 3 is definitely low need for fungicide or biofungicide agents to help bolster the crop's ability to deal with pathogens. Field 2 is low need, but it has a little bit of a hot spot here where maybe, in cases where there is a capability to do precision planting, or a planter that has the capability of a dual seed compartment to ultimately be able to switch up and plant a seed that is coated with biofungicide or fungicide. This is an area where maybe that would be helpful to kind of pinpoint that area in the field that has lower levels of these beneficial fungicidal microbes. And again, this northwest quadrant in field 1, I would say field 1 kind of falls in the moderate need category here, as a field to prioritize placing fungicide or biofungicide, because again, it just doesn't have quite as high of levels of natural fungal disease suppression as fields 3 and fields 2. So this will be, I think, our last disease example is Goss's wilt, which I believe is a pathogen that's known to overwinter in crop residue on the surface of the soil, and it's at lower levels than Fusarium. But looking at some of these fields, I think actually a couple of these might have gotten moved around a little bit. But just looking at the map, you can see in this field, I think this is field 2, but it's labeled here as field 3. But you can see a hot spot here of the Goss's wilt, in this sort of this eastern midpoint of the field. Field 3, I believe, up here, you can see a lot of gray, and in the gray, that's areas where the disease is not even detected. And similar for field 1 down here, it was detected at very low levels across quite a bit of acreage, but generally there's not really any hot spots. It's really this field right here that has a hot spot for Goss's wilt. And I know there are some seeds that have some Goss's wilt tolerance. So field 2 would be the one in this case to maybe prioritize seeds that have that Goss's wilt tolerance. Whereas field 1, field 3, it's good to be aware that it's present, but it's not necessarily a significant issue. So that would be a lower priority as far as seed placement. Now to look at insects, I'm going to try to run through this pretty quickly for the sake of time. In this case, we're not looking at insect levels in the soil. That's not necessarily something that Biomaker's technology is able to measure, but we're looking at the level of those biocontrol agents, specifically insecticide agents, so the beneficial microbes that are able to suppress insects, particularly insects that have some stage of their life cycle in the soil. So these might be microbes that are parasites of insect larvae, or microbes that excrete certain enzymes that repel certain insects. So these insecticide agent microbes, you can see they're pretty spotty throughout these fields. The average for field 3 up here is 47. So it's around the midpoint, 50, the 50 BeCrop score is right in the middle of medium. So it's about the mid-range scored back against Biomaker's database. So there is quite a bit of variability in this field, but on average, most of the acreage, if we look at the histogram here, falls in that medium range. Field 2 is quite a bit lower. If we look at the histogram, most of the acreage falls in the low range and the very low range. You can see that represented by how much yellow there is, and the average score is 31, so quite a bit lower. And then field 1 on the bottom right here is right about in the middle. There's quite a bit of yellow, also some hot spots. So this is a good example of where you might have quite a bit of difference in terms of the amount of insecticide or bioinsecticide, or even the BT seed traits that are placed in these fields. Certainly a precision planter situation could allow you to fill in some of these spots in field 3 and field 1. Some of these yellow spots that have low natural insect suppression, maybe that would be places where you switch to plant a BT seed or plant seed that's been treated with a bioinsecticide or insecticide. But field 2, this is a field where you'd want to prioritize some of these, either BT seed traits or insecticide and bioinsecticides across the vast majority of the field, since most of it is scoring pretty low. Therefore, there's not really a lot of natural disease or natural insect suppression going on. So field 2 is definitely the one where it would make sense to prescribe some of these tools to help deal with insects and just give the crop more of a bolster its ability to deal with some of those insects. Whereas field 3, field 1, a moderate need because the scores aren't super high. There's a lot of yellow, but there's still, especially field 3, there's quite a few, generally decent levels of these insecticide agents, since overall it scores about average, close to 50 when it comes to its comparison to Biomaker's database. Now looking at stress tolerance. So in this case, we're switching gears, and instead of looking at disease or insect related seed and seed treatment placement, we're talking stress tolerance. So obviously this is a case where context is a huge factor, whether or not fields are irrigated, whether or not they're in a drought prone region, or maybe a region that has a lot of environmental variability versus a more stable climate. That's really going to have a big influence here. But another factor that I think it's a really underrated aspect of soil health and soil biology is the fact that a lot of beneficial microbes can help crops deal with some of those abiotic stressors by producing phytohormones, like in this case, ACC deaminase we're looking at. This is a phytohormone that crops naturally synthesize, but microbes also produce. And when microbes are microbes that can produce ACC deaminase are present at high levels, what they're able to do is ACC deaminase essentially helps helps cushion some of those negative effects of stress. It essentially helps reduce the influence of those stressors on the plant, helps reduce senescence due to excessive dry conditions and soil salinity and things like that. So in this case, field 2 is actually pretty adequate when it comes to ACC deaminase producing microbes. We see most of the field is in the medium range, overall score of 45, so it's close to that, maybe slightly below the middle range of the database, but field 3 scored an average of 36, and most of this west half is in the low range. Field 1 scored a 35, so again, it's sort of in that about the lower third of BioMaker's database, and a majority of this field is in the low range. So this is a case where these two fields might be better candidates, higher need to prioritize either stress-tolerant seed varieties or biostimulant seed treatments that make claims of helping promote the crop's ability to deal with stressors like drought. So in this case, field 3, field 1 are those high need candidates to prioritize those seed varieties or seed treatments. Field 2, more of a moderate need, where maybe if it economically makes sense, or if there's maybe if this is in a region that's very susceptible to abiotic stressors, maybe field 2 is a candidate. But if this is in maybe a more stable climate or region, maybe field 2, you already have fairly solid levels of microbes that can help produce a vital stress response phytohormone. So field 2 is probably lower priority than field 1 and field 3. Next, and I think this is the last specific seed treatment and seed placement case study to look at. This is focused on placing biofertilizer or mycorrhizal seed treatments. Mycorrhizae, I want to mention, have a lot of great beneficial functions, not just on nutrient cycling, but also on stress tolerance and other beneficial and biocontrol and disease protection. But in this case, we're going to focus on how mycorrhizal seed treatments can be applied to improving nutrient cycling. Specifically, here we're looking at phosphorus cycling levels. So in this case, there's quite a big difference across these three fields. We're looking at the levels of microbes that support phosphorus solubilization, so the microbes that are able to unlock phosphorus and convert it into plant available forms. And field 3 up here, we have pretty adequate levels, a score of 56 out of 100 on the BeCrop score index. Maybe some spots in the east that are a little bit on the lower end, but overall phosphorus cycling microbes are above average here. Whereas field 2, the average score is 37, so below average. Almost the entire eastern portion of the field is quite low, whereas this northwest portion is a little bit higher. So field 2 definitely could benefit from biofertilizer or mycorrhizal seed treatment. Mycorrhizae are able to both solubilize phosphorus and help transport it to the plant for uptake. And then field 1 here scored the lowest at a average phosphorus solubilization score of 28, so quite low. So field 1, field 2 here really would be the strong candidates, the high need to prioritize biofertilizer and or mycorrhizal seed treatments. Field 3, actually I think these got mixed up a little bit too, but this field 3 here that scored a 56 on the top, maybe a little bit lower priority. It's in a case where phosphorus is a significant, phosphorus deficiencies are a significant issue, maybe prioritize it. But overall, this is a case where if we're looking at, which fields really can benefit from those seed treatments, we're going to go with field 1, field 2. And then lastly, and actually for the sake of time, I'll probably gloss over this one, but this is just showing the biodiversity index. So instead of looking at really specific microbial functions that we just looked at, like nutrient cycling, biocontrol, disease, this is just looking at sort of the overall soil biology status. And we can see that these field 3, field 2 scored around 50, so close to average. Field 1 down here scored a little bit lower, 39. And I know it's kind of a controversial topic on whether or not, biosolutions like biofungicides should be placed or be used to essentially, replace conventional fungicides and fumigants that are known to have some potential negative effects on the soil microbiome. So that's going to depend on a lot of different variables. But if you're just looking at which fields sort of have the, can use the most boost to the overall soil health and soil biology, field 1 here scored a little lower with 39. So this would maybe be a case where if you're weighing the pros and cons of biosolutions versus conventional crop inputs, biosolutions maybe here could help move the needle in this first field, just kind of help get the overall soil microbiome and biodiversity moving in the right direction. So with that, there's a lot more details there that I probably could have gone through, but I was trying to be light on time. So to summarize, this is sort of the rundown based on those 7 or 8 different case studies we just went through. If we're to combine them and kind of put them on a checklist, these are the prescriptions of seed type or seed traits and seed treatments that we would come up with. Again, going through, I'm not going to go through all the slides again, but field 1 could benefit from stress tolerant seed traits or seed treatments. So that could be, maybe prioritize seeds that are known to have vigor and deal with drought effectively. Biofungicide seed treatment to deal with some of those disease issues. Mycorrhizal seed treatment to help with nutrient cycling and just overall soil health. And biofertilizer seed treatment. Actually, field 1 was this one down here on the right that was a little bit lower on the biodiversity index. So that's why these 4 different seed and seed treatment products are all biosolutions, you could say, aside from stress tolerant seed traits. But that would be the case here with field 1, to prioritize a program that helps with disease, nutrient cycling, and mycorrhizae also are known to have a lot of great benefits on overall soil health. So that's sort of the prescriptive checklist here. Field 2, Goss's wilt had a hot spot there, so a Goss's wilt tolerant seed would be ideal to place. Maybe if possible to factor in or stack a BT traded seed, BT trait to help deal with, because there wasn't really high insecticide agents. So if there are insect pest issues in this field, that could be valuable. Biofungicide or fungicide seed treatment to deal with disease and help bolster the crop's ability to really fight off some of those fungal pathogens. And then biofertilizer seed treatment, again, mainly to focus on that phosphorus cycling deficiency that we just looked at. And then field 3, stress tolerant seed trait, because it was a little lower there. BT traded seed to help deal with any insect pests, and then biofungicide or fungicide seed treatment. But again, I'm not going to go all the way back, but field 3 was the one that had fairly adequate phosphorus cycling. So biofertilizer that was recommended for or prioritized for field 1, field 2, maybe not quite as much of a priority here. Maybe instead focus more on stress tolerance, insect resistance, and soil-borne pathogen resistance. So this was a really generic example. There's definitely a lot more depth you can go into with BeCrop data, but this is just a quick rundown on how BeCrop indexes can be used to really demystify and place seeds and seed treatments in what's obviously a very crowded category that also, I would say, seeds and seed treatments as far as bang for the buck and influence on yield and crop outcomes, probably one of the most important decisions, and also one of the opportunities to really get beneficial biology right where it's needed on the seed. So those variables, those BeCrop indexes we just went through, very important factors in ultimately making some informed decisions on seed and seed treatment. Lastly, looking beyond seed treatment, some other areas BeCrop Farm can be used. This is another feature in BeCrop Farm which offers a recommendation, or it's called best fit solutions. So using Biomaker's database of actual replicated trials, this interface it basically gives you insights onto which biostimulants are projected to perform well in similar, in this specific soil type and environmental profile. So in this particular, these are the same 3 fields we were just looking at, and a couple of these fields are recommended to receive bacillus-based biostimulants. And then this field in the bottom left is recommended for trichoderma-based biostimulant, again, based on the soil genomic data we just looked at, as well as the environmental data, soil type and things like that. So I'm not going to get too deep into this for the sake of time, and this will be a topic that we'll touch on in upcoming Biomakers webinars to go through some of the other ways that BeCrop can be used to place products and inputs. So with that, I wanted to make sure we save at least a little bit of time for questions. So sorry, I did want to stick more to like 10 or 15 minutes, but we'll try to answer as many questions as we can. And then any questions that I can't get around to, we'll reach out to and get some answers to you, connect you with the Biomakers team if necessary. So just looking through a couple of the questions we received now. So this is an interesting question, kind of not really used to. I'd say something that, I don't have a lot of key insights or experience with, but the question is, electroconductivity, soil mapping, and soil testing in identified areas are challenging in that an identified area may not be large enough to effectively manage with large application equipment. And I do see this a lot in the specialty crop space, and in certain cropping systems, it's not always not always efficient to be using large equipment for applications, and it can be also sometimes hard to key in and really look at variability across the field if the field's only maybe like 5 or 10 acres. Do you have a solution for micro precision crop management? And one of the factors I'd mention here is something that I think is gaining a lot of steam. I've seen at a lot more ag events is the use of drones to provide what I'd call micro precision crop management. So drones, and then also I've seen autonomous spray rigs. I don't remember any of the trade names or companies off the top of my head that offer these, but these are basically remote controlled or self-autonomous sprayers that can fit in very narrow rows, say in like a vineyard or a specialty crop setting, that can be used to spray, where tractor mounted spray rigs can't fit. So drones and some of these autonomous spray rigs would be solutions there. And in cases where BeCrop Farm does sort of have a minimum acreage, in some cases of maybe smaller field, maybe only add up to 10 or 15 acres or less, using individual BeCrop tests is always a great solution there, where you can look at the same sort of data that I just went through, but on an individual test report basis, so like a standard soil test, and that can be used really in the same sense to inform some of these same decisions around seeds and seed treatments. So next question, what is the typical turnaround time on the test results? So I'll give a general range, but I'll have to defer to the Biomakers team for specifics of, because there can be some variability depending on how busy the lab is. But generally, turnaround time is within 2 weeks. I'd say 1 to 2 weeks. Can sometimes be faster, but again, can vary a little bit based on factors like how busy the lab is. But I would recommend getting with a Biomakers account manager to get down to the specifics and also discuss any other factors that might affect turnaround time, but generally in that under 2 week range. Let's see. So we only have a couple minutes left. There's some really good questions here. So this question asks, can you please tell me how you do cross-validation of the soil biodata, for instance, to filter out necromass? So Biomakers has a section on their website called Tech Validation, which does have some of the peer-reviewed research papers that sort of went into informing the testing process and data analysis process. But my understanding is that there has been, so there is a, I'm trying to think of how to explain this in a very, very brief, succinct manner. So on the question of microbial necromass, there is a technique that allows the necromass to be filtered out, and basically that's an enzyme or a compound that's added, which binds to and removes any microbial cells that are bursted. Because when microbes die, when their cells typically will burst, and then that DNA kind of spews out. So there's a method that can be used to filter out all of that necromass so that there's not any contaminant or any non-living DNA there. But Biomakers actually did some internal work to look at whether or not the necromass had a big influence on the soil microbiome by analyzing samples that had the necromass filtered out and samples that didn't have that dead biomass filtered out. And what was found is that necromass tends to break down pretty quickly and doesn't actually have a huge influence on the results. And in fact, microbe populations turn over so quickly, that necromass actually, you know, the little bit, that small percentage of dead biomass that actually is measured can actually give you a little bit of a wider temporal fingerprint, or wider, you know, wider range of understanding of what microbes were present. Because even if a microbe is just recently died, you know, it had an influence on the soil microbiome. You know, it was there for a reason. So that necromass actually in biomakers technology is not filtered out, and that's intentional, because it is shown that that sort of that leads to a little bit stronger outcomes. And as far as the, you know, the cost and you know, the labor of going through that filtering process, it doesn't really make the results any more robust. But it is something that if a client does want to talk to biomakers about, you know, introducing that step in the process to filter out necromass, that's something that can be discussed with biomakers team on a case-by-case basis. So yeah, that's just one example of the cross-validation that's gone into biomakers data, but they've also done some of those research papers I mentioned in the tech validation page have looked at other factors like yield and soil fertility testing and crop measurements to cross-validate and determine how the soil microbiome is shaping and influencing those factors. So I won't get into the details of any of those research papers now, but they're available on Biomakers' website and can provide a little bit more insight on how the biodata has been cross-validated with other factors. Lastly, let's see, what time of year do you like the testing to be done? That's really a question that depends a lot on crop cycle. It's one of those cases where, you know, the soil microbiome, the microbes are always going to be there even in the winter. They're just going to be dormant in the winter, obviously, when it gets below freezing. As a best practice, Biomakers does not recommend sampling when the soil is, you know, the soil surface, or at least the top 6 inches is frozen. So generally, ambient temperatures ideally would be above, you know, 0 Celsius, or about, you know, 38, 40 degrees Fahrenheit. But as far as actual timing, what I recommend is taking those samples at the time that is most pertinent to informing management decisions. So taking samples, you know, again, that turnaround time is roughly, you know, 2 weeks, maybe allow 3-4 weeks buffer to be able to have some time to make that decision. So I would recommend pulling samples roughly a month, at least a month before a major decision, or before agronomic insights are needed. So it really can be done throughout the season. It can be done after harvest, before planting, even, and in season is probably the best sort of time to really check on the soil microbiome, because that's when it's actually interacting with the crop. So really, the sample timing is very versatile depending on, you know, what the goal of the samples is. But the #1 rule of thumb is it's hard to get a soil core in the ground when the soil is really frozen over, but also when that soil microbiome is dormant, it might not provide the best representation of the soil microbiome during the growing season. So best practices to avoid during the really cold times of the year, but generally across the growing season, and as well as leading up to and after, you know, leading up to planting and after harvest, those are all, you know, valid times of the year to be doing sampling. With that, we're a couple minutes over, so I'm going to conclude things here. And I know we did get quite a few other questions we'll be sure to follow up on. Again, this webinar will be recorded and be available for anyone who wants to rewatch it, or if you weren't able to tune into the whole thing, you'll have the whole video at your disposal. I want to thank you all for joining in and you know, joining me to review some of the practical uses of BeCrop technology alongside to inform placement of seed treatments and seed traits. And look forward to contacting, reaching out to anyone who's interested. And yeah, thank you for tuning in. We'll be sure to be in contact, and best of luck using BeCrop technology to make the best soil health decisions. And ultimately, you know, appreciate you for being interested in some of these cutting edge technologies that are really helping push the regenerative ag movement forward. Thank you, and have a great rest of your day.